Welcome back, my dear students. Today we are going to explore one of the most widely used immunological techniques in research and diagnosis, that is the enzyme-linked immunosorbent assay, or simply ELISA. This method revolutionized how scientists detect and quantify hormones, proteins and antibodies without using any radioactive substances like radioimmunoassay. So let's dive deep into its principle, type, types, applications, and even its advantages and disadvantages in a simple and clear way. ELISA basically works on a very simple but powerful principle, the specific binding between an antigen and antibody. This is the image showing the binding of antigen and antibody. In this technique, antibodies are linked to enzymes instead of radioactive levels, as in radioimmunoassay. When the antibodies bind to its specific antigen or hormone, the attached enzyme catalyzes a color producing reaction. The intensity of the color produced is directly proportional to the amount of hormone or antigen present in the sample. The color change is then measured spectrophotometrically, giving a quantitative estimation of the analyte. So in short, we can say that more color is equivalent to more hormone concentration. This is the image showing different types of ELISA like direct ELISA, indirect ELISA, sandwich ELISA and comparative ELISA. We'll study in detail about all the types of ELISA. Now, firstly, we should to know the basic process, basic step-by-step process of the ELISA. First step is the coating. The antigen or antibody is first immobilized on a solid surface, usually a 96-well microtiter plate. Blocking the remaining unbound sites are then blocked with a protein solution like bovine serum albumin or BSA to prevent non-specific binding. Then binding the test sample containing target molecule, whether it is antigen or antibody, is added, allowing it to bind the immobilized partner. Then enzyme conjugation and enzyme linked antibody is added, which binds specifically to the target molecule. Then substrate addition, a colorless chromogenic substrate like TMB or OPD is added. The enzyme converts then it into a colored product. Detection, the intensity of the color is then measured using a spectrophotometer or ELISA reader, giving a quantitative result. Types of ELISA, there are basically four types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA and comparative ELISA. Direct ELISA. Direct ELISA is basically the simplest and quickest of all other types of ELISA. A single enzyme-linked antibody is used which directly interacts with the antigen present in the sample. In direct ELISA, the antigen is immobilized directly on the plate. Then a single enzyme-labeled antibody binds to antigen. After washing, the substrate is added and a color is produced. Advantages: quick, fewer steps or less chance of cross reactivity. Disadvantage: less sensitivity since only one antibody is used. The direct ELISA is used for detecting antigens like certain viral proteins. Direct ELISA procedures: addition of sample containing antigen to the well of the microtiter plate, then antigen gets adsorbed to the surface of the well, then washing it to remove unbound antigen, the addition of enzyme linked antibody that with the antigens if present, washing to remove unbound antibodies, then addition of chromogenic substrate, visualization of color change and result interpretation. Direct ELISA application, it is used in identification of biomolecules and also for diagnosis of infections of Mycoplasma bovis. This is the image showing the steps of direct ELISA. Indirect ELISA, it is the most popular type of ELISA in use. Antibody detection is carried out here in indirect ELISA. A secondary antibody is linked with the enzyme is used. This process was basically developed in 1978. The antigen is immobilized first like direct ELISA, then a primary antibody, unlabeled antibody binds to the antigen, then a secondary enzyme linked antibody binds to the primary antibody. What is the advantages? It is more sensitive because multiple secondary antibodies can bind to one primary antibody and amplifying the signal. Commonly used for detecting antibodies in a sample like HIV or hepatitis antibody testing. Indirect ELISA procedure: addition of known antigen specific to the antibody of interest, careful washing to remove unadsorbed antigens or some adsorbed contaminants, addition of serum samples to their respective wells. If there is presence of specific antibodies, they will bind to the antigens. Was again an addition of enzyme-linked secondary antibodies to the wall. Was again addition of substrate. Visualization of color change and result interpretation. Indirect ELISA applications, it is used for the detection of HIV, rubella, dengue viruses, etc. to detect. It is also used to detect certain drugs in serum sample. Sandwich ELISA. This process was developed in 1977. In this process, two different antibodies like capture antibodies for attachment sample antigen and enzyme-linked secondary antibodies are required. The antigen of interest first binds with the capture antibodies and then secondary antibodies bind with the epitope antigen, forming a sandwich like structure with antigen in between antibodies. In this process, in this step, in this sandwich ELISA, the plate is coated with a capture antibody and the antigen from sample binds to this antibody. Then a second enzyme linked antibody binds to another epitope on the same antigen. The advantages are extremely specific and sensitive. It works even when antigen concentration is very low. Used for measuring hormones like insulin, FSH, LH, HCG and cytokines in serum or plasma. Sandwich ELISA procedure: addition of capture antibodies in microtiter well, washing, addition of serum sample like containing antigen in the well, washing, addition of enzyme linked antibodies in the well, washing, addition of substrate, then visualization of color development and result interpretation. Sandwich ELISA applications involve mainly, it is mainly used for detection of rotavirus and enterotoxins of fecal E. coli. Then pregnancy test kit are based on this technique of sandwich ELISA. Competitive ELISA, it is the basic concept of this type is that there are competition of binding occurs between the sample antibodies if present and enzyme linked secondary antibodies to the antigen. If the sample contains specific antibodies of interest, they will bind to antigen and won't allow the enzyme linked antibodies to bind with the antigen, which are then washed away. Hence, after the addition of substrate, there won't be a color change indicating a positive test and vice versa. Competitive ELISA was developed in 1967. Here the antigen in the sample competes with a known enzyme labeled antigen for limited antibody binding sites. The higher the sample antigen concentration, the less color is produced. So it's inversely proportional to concentration. It is used for small molecules and hormones like steroids, thyroid hormones and drugs. Competitive ELISA procedure include addition of HIV antigens in the microtiter well, then washing, which is followed by addition of serum sample, and again washing, and then addition of enzyme-linked HIV specific antibodies, then again washing, and then addition of substrate, then visualization of color change and result interpretation. Competitive ELISA application include, it is mainly used for detection of HIV. ELISA result interpretation, quantitative interpretation, that is the presence or absence of antigen, is done by visualizing the color change in the solution. If the antigen antibody reaction takes place and bound enzyme linked with antibodies remain in the solution, they will modify the substrate and color change can be seen. Quantitative interpretation, that is the concentration of antigen or antibody, can be identified by measuring the optical density of the solution with the help of ELISA reader spectrophotometer. The optical density versus concentration curve is prepared for both the standard sample and unknown sample and compared to determine the concentration in the unknown sample. Applications of ELISA. ELISA is essential diagnostic and research tool. Some major applications include hormone assays like detection of insulin, follicle stimulating hormone, luteinizing hormone, human chorionic gonadotropin and thyroid hormones. Disease diagnosis, HIV and hepatitis B or C antibody testing, COVID-19 serological testing, pregnancy test using HCG detection, allergy testing, detection of bacterial or viral antigens, and then food and environmental testing, etc. The advantages of ELISA: it is safe, Here is no radioactivity unlike RIA. It is economical and easy to perform. High throughput, many samples can be tested simultaneously in this procedure. Automation friendly, modern ELISA readers and robot make it efficient for clinical labs. Disadvantages of ELISA include lower sensitivity than radio immunoassay or RIA. Enzyme stability is crucial because temperature and pH can affect the results. Requires optimization of reagents and incubation times. Cross reactivity may occur if antibodies are not specific enough. So my dear students, today we have learned how ELISA provides a powerful, safe and versatile method for detecting hormones, antibodies and many disease markers, all through the clever use of enzyme-linked antibodies and color-based quantification. Whether in medical diagnostic, research lab or public health screening, ELISA remains one of the most trusted immunological tools. If you enjoyed learning about ELISA and its types, don't forget to like, share and subscribe to my channel for more clear and engaging biology lessons. See you in the next video where we continue exploring the fascinating world of biochemistry and immunology.